I'm just gonna sit here for a second because I'm still like, as to what just happened. Sorry, I will finish going through some of the other 7th grade stuff real quick. I had to take that phone call, and I don't know how to like pause live streaming. I don't think that's an option. Nope, not an option. So here we go. So we were talking about cells. What about cells? Don't really remember, just cells. I know the next question was asking about why do we think that the heart is important, or why is the circular system the most important? Yeah, since the circulatory system pumps blood throughout the body, why might one think it is the most important of the organ systems? All parts of our body depend on the circulatory system to bring them new oxygenated blood. Is it because the circulatory system restricts blood flow to other parts of the body when it wants to do so? The heart is the biggest organ in the body, and the heart is not the biggest organ in the body. Remember, your heart's only about the size of your fist. Blood pumps only in the most important areas of the body. All areas of the body are very important. Your answer should be all parts of the body depend on the circulatory system to bring them new oxygenated blood. So that was survivor inside the cell membrane. Now we're looking at cells as the building blocks, or the body's building blocks. Let's scroll down. So in this section, we talked about plant cells and their cell structure, the cell processes, cell division, the cell's formation of tissues, and how tissue forms organs. So that should be a review for a lot of the 7th grade that's doing high tech. All cells are enclosed by the cell membrane. I'm going to turn off my phone to jump. Okay, now that that's taken care of. What is a unique function of muscle cells? Is it muscle cells don't transport water, and they don't have the ability to empty. They also do not have the ability to pump blood. So the ability to empty is your bladder or your bowels, and then transport water would be the circulatory system. Ability to pump blood. The ability to pump blood would be the heart. So the unique function of muscles is the ability to contract and relax. All of the following are types of digestive system organs, except that one should be pretty straightforward. So you have intestines, heart, esophagus, and stomach. The only thing that's not part of your intestines helps push through digestion. Esophagus is here in your throat. The stomach digests your food. The only thing that's not part of your digestive system is going to be the heart. With shipwrecked, I don't think there are questions that we have to answer. Oh. Oh, no, there are. So drinking seawater has what effect on the body? Seawater is very salty. So we wouldn't want to drink seawater. It's just because it's going to make you very dehydrated. So the effect of seawater leads to dehydration because of the salt. Which part of the body has trouble dealing with the seawater? The skin, the nervous system, the eyes, the digestive system. Your skin, your nervous system, and the eyes are all going to be affected because of the seawater, because it begins to dry you from the inside out. So the one that's going to have the problems to start off with will be the digestive system. Last question for this one. Uh-oh. I did check email. There it is. What is not a possible effect of dehydration? When you're dehydrated, you're not going to get hungry. but everything else is going. You can die from dehydration. You're going to hallucinate because the blood is very thick in your system, and you'll also get cramps. Your muscles won't have the proper nutrition. So, but what you won't feel is hunger. You'll be very thirsty, not hungry. So a fish's kneecaps. Mr. Kneecaps, are you part fish? Scanning electron microscope. It's okay, I have to be right back. Da-da-da. The acronym SEM, what does that stand for? It is SEM. So it's in the paragraph 2 right above. It says the scanning electron microscope is capable of enormous magnification. I don't know if that's something that you should be bragging about, drinking seawater with just a little bit of fish poop. So the acronym SEM stands for Scanning Electron Microscope. Be very careful when you're clicking, apparently. Oh, I clicked the wrong one. So the first one says Scanning Electric Microscope. You're looking for Scanning Electron Microscope. Which of the following criteria must be present for a light microscope to work? The object is thick enough for light to pass through. The object is thin enough for light to pass through. The object is thin enough for light not to pass through. The object is thin enough for darkness not to pass through. So if we want a light microscope to work, light is going to have to be able to pass through. In order for light to pass through, the object has to be very thin. They're busy with schoolwork. Thank you so much, Kneecap. So are you related to Gaster? Number 3. What is the major difference between a light microscope and a scanning electron microscope? Oh, we're going to have to go read for that one. I'm not 100% sure. Okay. So is the scanning electron microscope much more sophisticated than a light microscope? The light microscope has an electric gun that fires a stream of electrons at the specimen. No, a light microscope is pretty standard. So it's just it'll have your base and then a light that usually shines up from the bottom. The light microscope is much more sophisticated than a scanning electron microscope. Again, no, it's pretty basic. The light microscope creates micrographs that can be colored by hand or computer to show more detail. The light microscope doesn't do that. You would put a slide on the light microscope to see what's going on with it. The scanning electron microscope is going to be the one that would be able to magnify and be colored on the computer. So number 3, you should be the scanning electron microscope is much more sophisticated than a light microscope. Oh, you're their friend? Okay. You have a question. Do crabs think fish are flying? Maybe. I mean, well, fish fly, don't they? They fly through water. Flying is kind of relative. Flying would be how fast something is moving. What do you think? How fast do you run? Or do you fly through water? Okay, I see a flaw. If you're a fish and you don't have legs, then why do you have kneecaps? You have does your tail regenerate? You stole them? Hmm. You, so are you a fish without kneecaps or so hold on. Are you a fish that doesn't have legs but other fish do? He was the last of his kind. You made a fish population go extinct. I don't think you can hang out with us then because you're not a nice person because you made a fish population go extinct. They make you look snazzy. Okay, I'm gonna go through maybe one more of the how cells reproduce, and then I'm gonna go ahead and jump off for the day, and then I'll be back on Tuesday with any other. Hello, Gaster, welcome back. With any other questions that might have happened or not happened with for the new assignments. Oh, okay. So cells reproduce. That is just collecting coins. Make sure you read the article, and then we're looking at um, Anton von Leeuwenhoek. Interesting stuff. So, what was the name of Robert Hooke's book written in? Yes, we're talking about cells today. The name of Robert Hooke's book written in 1965. Oh, Micrographia. So it's in that first sentence at the top of the page. So it's called Micrographia. Why was von Leeuwenhoek's why was von Leeuwenhoek's magnified lens better than Hooke's? Probably because it magnified better. So Hooke was only able to magnify objects by a power of 30. Where is it? Kneecaps, is your name really Nemo? Are we needing to go look for you in Australia? So again, Hooke was only able to accomplish having a microscope magnified by powers of 30, whereas von Leeuwenhoek was able to get it to 275 times. Why would the Royal Society be skeptical of von Leeuwenhoek's findings? Usually, when, especially in that time, the Royal Society or just society in general would be really skeptical of scientists' findings because it seemed too good to be true. So, looking up, it's in the second paragraph. So it says, for 50 years, he sent letters to Hooke and other members of the London's Royal Society that showed his findings. He wanted to share what he had learned, but von Leeuwenhoek was secretive about his methods and reluctant to share his tools with others. Von Leeuwenhoek was the first to observe bacteria and protists. The single-celled organisms were unknown at the time, so they were skeptical. So the skeptical Royal Society sent a team to observe von Leeuwenhoek and evaluate his work. The team saw that his findings were accurate, which opened up a new world of exploration for decades to come. So it wasn't because he couldn't back up his research, and it wasn't because he lived in France, and it's that he never proved how he got his information because they observed him. It was because he observed something that wasn't known at the time. And again, typically, if you were the only person that saw something at that point in time, other people didn't believe you about it. So that takes you through really the majority of the assignment for Studies Weekly on week 2. If you have any questions, please make sure that you email me. And then I will be back on Tuesday for next week's assignments as well. So I'll see you then. Bye.